Hello, my name is Bert Kosiski. I am president of the Board of Trustees of cureCADASIL. And on behalf of myself and my colleagues, and we have Benita Ball, who you cannot see, but she is listening in and helped organize this webinar setup, and also Jane Gunther, who is also a trustee and past president and our science person for cureCADASIL. So we all like to welcome you to the CADASIL Connection webinar series. And we're very fortunate to have Professor Dr. Helena Karlström, who is going to speak on immunization therapies for CADASIL. But first, just a word about our organization. cureCADASIL is a nonprofit organization dedicated to helping patients, families, caregivers, and other supporters touched by CADASIL. We aim to unite patients and the medical community toward common goal of treatments and ultimately a cure for this rare genetic disease by providing communication, advocacy, research, and education. So Dr. Karlström is head and research group leader at the Division of Neurogeriatrics, Department of Neurobiology, Care Science and Society at the Karolinska Institute in Stockholm. She has made important contributions in understanding the mechanisms in Notch3 biology and CADASIL. And most recently, her group has developed an active immunotherapy project with encouraging results. Dr. Karlström is the grant recipient of the 2023 $1 million bike ride, which funding was from February 2024 through January 2025. And I might add that the $25 million bike ride is coming up here on this upcoming Saturday. She investigated ways to restore the brain vascular system in a CADASIL mouse model that has an ongoing pathology characterized by aggregation or clumping of Notch3. She has investigated two strategies, active and passive vaccination against Notch3. She has shown that immunizing the mouse model against Notch3 reduced the aggregation of Notch3 in brain and small blood vessels and levels of Notch3 in the blood. The latter suggesting this could be used as a potential biomarker. If these results can be replicated in humans with CADASIL, it could lead to important new therapies. So before I hand it over to Dr. Karlström, I'll remind everyone to note that the audience will be muted during the presentation, but we'll take questions at the end. So if you have a question, if you can type it into the question box in the drop-down menu shown here on this slide, that will be appreciated, and we'll get to them at the end of the presentation. So I'm going to hand it over, hopefully things work, to Dr. Karlström and see if we will be able to see her screen.
So, do you see my screen?
Yes, looks good.
I can see if it works with the presentation mode as well. So otherwise I go back and just show it like this.
Yeah.
Is this?
I think it looks good.
Working okay? Okay.
Yes.
So thank you, Bert, and thank you everyone for having the opportunity to present my research about the immunization therapies, and also thanks to cureCADASIL for the great opportunity to use the grant for these projects. So what I want to talk about today is what we have done a bit, but also what we are doing, or what is ongoing in my lab in order to find treatments for CADASIL. We'll just start a bit with the background of of CADASIL, and I think everyone is aware of that. It's a short name for a long name. It's cerebral autosomal dominant arteriopathy with subcortical infarcts and leukoencephalopathy. And as we also know, it's the most common monogenic form of small vessel disease. And it's caused by mutations in the Notch3 gene, which is leading to Notch3 extracellular deposits. And Notch3 is an important protein which is expressed in the brain vessels, especially in the arteries and in the small capillaries. And as you can see on the right-hand side here, that's a schematic figure of how the brain vessels looks like in the bigger arteries, which then is going down with the small arterioles in the brain parenchyma and ending up in the smaller vessels called capillaries. And these brain vessels are, of course, very important in order for supplying the brain with oxygen and nutrients, and also the arteries and capillaries are very important to also regulate the blood flow and so forth. CADASIL, it's a rare disease, but it has been shown in different studies, in genomic wide studies, that the Notch3 mutation can actually occur in quite a large population, although the symptoms can vary quite a lot. So that's why it's also a bit underdiagnosed. And as we know, there is no cure for CADASIL so far. So Notch3, as I said, is an important receptor which is expressed on the vascular smooth muscle cells and also the pericytes, which are the main cells in the brain vessels. And what is happening is when we have a mutation in Notch3, which are leading to CADASIL, it's that the mutation causes a conformational change of the protein. So if we have a non-mutated Notch3 protein up here in the circle, a purple circle, and if we have a mutation, this protein starts to look differently, and they also start to clump together and aggregate. And this is also recruiting other proteins around the vessels. So they also start to aggregate together with the Notch3 mutant protein. So this is just to show how it can look like on the brain vessel. We have these clumps or aggregates, and including then Notch3, but also other proteins. And this can be visualized by different techniques, but MRI is a very common technique to use in the clinic, where we can see that it starts to be small holes or lacunes, and the white matter changes. And this will be a bit of more detail and molecular, but I just want to show that Notch3, which is this gray and the purple one spanning a cell membrane here in yellow, it's expressed on the vascular smooth muscle cells in the brain vessel. And normally this receptor or this protein binds to another protein which is on the adjacent cell. And what this then happens, it leads to, is that there are specific events like processing and cleaving events happening in the Notch3 protein, leading to liberation of the intracellular domain, which goes into nucleus, and it's very important to activate other genes. So this is the natural way of Notch3 processing and signaling. However, when we have a mutation which is occurring in this gray part here, then this signaling or this processing is happening, but the extracellular domain is not taken up by the other cell, which is occurring when we don't have a mutation. So the mutated extracellular domain, it stays here between the two cells and it starts to aggregate. And on the right-hand side here, we can also see these. We can also see these mutations occurring where they are located in the extracellular domain. And the higher the peaks are here, the more variants exist in this specific location of the protein. And if we just zoom in on one of these small little gray boxes, we can see there we have 6 small blue circles, and they represent an amino acid called the cysteine. And normally we have 6 cysteine residues in this small little gray box called EGF repeat. However, when we have a mutation, we either get an extra cysteine, so we will get 7, or we lose a cysteine, so we get 5. So this means that we have an odd number of cysteine residues within this little gray box. And this is what is causing the protein to aggregate. And this is just a highlight within this region. There are the majority of mutations happening here. And also it seems like the mutations which are more towards the left side in this schedule seems to be more severe than the other mutations which are more located more to the right of this domain. So our hypothesis is quite straightforward. We believe that the CADASIL is caused by these Notch3 extracellular domain aggregation, and clearing of these aggregates will rescue the vasculature to its normal function and improve or abolish the symptoms for CADASIL patients. So we believe that these aggregates are actually causing the disease, and that's what I think most of the researchers also believe. So it's not a very strange hypothesis. However, the way to remove this can be different approaches. And our approach is to use immunotherapy to clear and to get rid of these aggregates. This figure, I just want to show how it looks like or what we can use in order to see the Notch3 aggregates or clumps in the vessel wall. We can use different techniques, like we use something called immunostaining with immunofluorescence on tissue or cells, and we use a microscope with very high magnification in order to see the vessels, but also the small dots here. If you can appreciate the red dots here in the middle, they are representing the Notch3 aggregates, the small clumps. And this is just a way to visualize this in another way, using different software techniques where we can actually see these small red dots in the vasculature. On the right hand side, we can even magnify a lot more to actually see on one specific cell. So this is one cell, which is the smooth muscle cell expressing Notch3. And you see the little star up here. These are the GOMs, which are the granule osmophilic dense material, which are the aggregates of Notch3, but also the other proteins which are recruited by the aggregated Notch3. So this is something we can use when we want to see if our immunotherapy has worked or not. So first of all, there are 2 ways you can use immunotherapy. We can either have an active immunization, and this is that we are using a protein or another type of small little peptide, which we immunize in this case our mouse model. And this is like we have been immunized by COVID vaccine, for instance. So what is happening then is that when we immunize this, the animal, in this case the mouse model, is evoking or producing antibodies against these aggregates. So this is what's happening in the mouse model. And the Notch3 antibodies, they found these Notch3 aggregates and start to bind and also recruits other immune cells, which are represented here by the yellow small little things, which comes and eat up the aggregates. So this is active immunization. Then we have something called passive immunization. So here we generate antibodies in the lab. And this, I will come back to that later, how we have done that. So we are generating and producing antibodies which binds to these aggregates. And then you can immunize the mouse model in this case with these antibodies, which are also searching for a target, which in this case is the NOTCH3 aggregates and clumps. And in the same way as active immunization, it recruits the immune system to come up and clear away and eat up these aggregates. We use a mouse model which have a mutation, and it's a CADASIL mutation. This is just to show you that there are two different strains. And first of all, we used a strain called the 150 strain, which is the blue line here. It has an onset of about 5 months, at 5 months of age. So remember that a mouse model can maybe live up to 24 months. So at 5 months of age, we start to see aggregates in the brain. And this is the model, and we can see at 6 weeks, maybe small things, but especially when they are 1 year, we can see quite a lot. And even at 20 months, we have a lot of NOTCH3 aggregates. So we use this mouse model in order to actually do a preventive study. So we want to immunize a model which has not got any NOTCH3 aggregates in the brain. So we want to come in before. So the first study we have done is to use this mouse model. And we started when they were 3 months of age, so 2 months before NOTCH3 onset. And this has been published, but I will just walk through the data because it's, I think, important what we have done and to then follow up what we are doing right now. So what we did is to produce a small little protein of the NOTCH3, not the whole protein, because that is a very big protein. So we produced that in a cell, and we purified it on different biochemical ways. And as you can see here, we run something we call a gel to see if we can identify the protein. And yes, we did. And we found that the protein was forming different sizes. So monomers means that they are just single proteins, dimers and trimers, they start to aggregate. So we can see that the protein starts to aggregate. So we took these aggregates and we injected this into the mouse model when they were 3 months. And then we continued for 4 months and give them some repeating booster. And what we can see already after 1 month is that the mice which got the NOTCH3 vaccine, which is the red lines here, they start to make antibodies against this protein. After 2 months, there was quite dramatic increase. And then after 4 months, when they were 7 months old, we had a very robust immune response in the mouse model compared to the mice which didn't get any NOTCH3 vaccine. And as Bert also mentioned, that we could see that the NOTCH3 protein was reduced when we had immunized it. And it is also increased from 3 to 7 in a non-immunized mice. So this could indicate that we can use this NOTCH3 in the blood as both a disease progression marker, but also a marker to see if the therapy has been efficient. But more importantly, we wanted to see how it looked like in our mouse model and especially in the brain vessels. So I have just circled around the main findings is that we could measure the number of aggregates in the small capillaries, in the small vessels, and also the size of the deposits. And we could see that both the number and the size of the NOT3 aggregates were reduced after immunization. So this looked very promising. However, when we are working with therapies and also especially with vaccination, we of course want to see if there are any side effects. And one way to look at that is to look into the retina in our eyeballs, because there we have a very, very nice vasculature, and we could see we can visualize the retina here, or the vasculature in the retina, in a normal mice, which we call the wild type. And then we had the CADASIL mouse model here at 7 months of age, which has not been immunized. And on the right hand side, we can see the vaccinated mice here compared to the ones which only got an empty buffer compared to the vaccination. And we didn't see any changes in the vasculature, because one thing that could happen is that the immune system starts to also eat up the brain vasculature, and this is not something we don't want to happen. And just to compare how it looks like in a mouse model where we have removed all the NOT3 receptors, you see a lot of gaps in the vasculature in the retina. And so this is also happening very early on when they are 3 months of age. So we didn't see any changes in the vasculature. Another way of looking at side effects is to look into other organs and also for inflammation markers. We looked into the kidney and we stained more histology staining to see if the morphology was looking differently between the vaccinated mice compared to the sham, which were not vaccinated. But also we looked for specific markers for cell death called apoptosis, caspase-3. And we didn't see any difference between the vaccinated and the sham mice. And also in the blood, we looked for different inflammation markers and also for neurodegeneration markers like neurofilament light chain. But we didn't see any differences there. So it seems like the vaccination was tolerable and safe in this mouse model. So this is something we have published a couple of years back. And then we got the funding from cureCADASIL and also from some other sources. And we could then start a new round of active immunization. We call it active immunization 2.0. And this is more a therapeutic treatment. The other one was a preventive treatment. We wanted to come in before NOT3 aggregation starts. In this case, we want to see if active immunization also can happen in a mouse model with an ongoing pathology. So that's why we are changing the mouse model from the 150, which was the blue one here, to the one which is black, called 350. And as you can see on these two graphs, the curve is quite dramatically changed. The onset is happening quite rapidly. So we wanted to come in when it was more or less has started to happen, when they were 3 months old. And we then wanted to continue longer than 4 months, like we did for the first study. So we wanted to do it for 9 months when they were 12 months old. What we also changed in this protocol is that now we wanted to be have a more pure protein to immunize. In the first active immunization, we used a mixture of both aggregates, but also the proteins which were not aggregated. And we also used a cell line which we culture in a petri dish. And this means that you need a lot of petri dishes in order to get a decent amount of protein. So we changed our way. So we did some new cloning into a different cell line, which is working more as suspension. And as you can see here to the right, this is quite a big chamber with a lot of cell media and a lot of cells which are swirled around, and we can then grow the cells when they are in suspension instead of when they were adherent to the Petri dish. And what we did here is to took all these cell media, and we started to purify it with the different biochemical techniques to see if we can remove the more monomeric forms, which is this big peak here. And on the right hand side, you can see that we have monomers, a large portion here, but we also have dimers and multimers. So different ways we use a different type of size exclusion chromatography, and we could separate these dimers and monomers from the monomers. So we have a more pure protein to immunize in our mouse model. So this is what we started with 1 year ago in May, actually. We started to immunize our more severe mouse model when they were 3 months old. We gave them boosters 9 times. We also collected blood during the whole period. And in February this year, we took all the mice. There were 20 mice which got the NOTCH3 aggregates and 20 which only got a vehicle. So we took them down, and we have now taken all the tissues, and we have taken blood and everything. But we also, of course, during this time, when we did with the immunization, wanted to see if the mice were affected in any way. And one way to do that is to look at the body weight, because that's quite an immediate sign if the mouse is not doing well. But we didn't see any differences between the treatment and the vehicle mice, which was good. And of course, we were very curious to see if we have evoked an immune response in the mouse model in the treatment group. And yes, we have quite a dramatic immune response here, showing in the red dots. So this was good. So now, of course, the main thing is to look into the brain vasculature to see if the NOTCH3 aggregates has been reduced. We want to see if we can find this blood biomarker, the NOTCH3 protein in the blood, but we are also looking for other biomarkers with different types of platforms and techniques, because we believe that maybe we need more biomarkers in order to see all the different stages, and also to follow the progression, and also to see if the immunization has been efficient. An easy way to actually look for efficacy of the therapy. And of course, we are very concerned about side effects as well, although we didn't see any differences in the body size, body weight of the mice, but so we will look into the different organs to do that. And the NOTCH3 deposits we will use for immunofluorescence staining on the tissue, and also we will look into this electron microscopy technique to see if we can count the number of GOMs on the vascular smooth muscle cells. And this is right now ongoing. I have a postdoc which I recruited in January, who has started to work on this full time. And as you can appreciate, there are a lot of mice, a lot of brain, a lot of tissue, a lot of blood to work on. And so we are also setting up new methods and techniques in order to do this in more multi-array scale, to do it more automatically. And we have used different antibodies to detect NOTCH3 deposits, and these are in red, in yellow here in the middle, we can see all the small little dots, and we compare the dots with where we can find the vasculature. The CD31 is staining all the vasculature, and CD13, which is the green one, is staining more the capillaries. So this is something which is ongoing right now, and so we will use a slide scanner to do this more automatically and start to measure the number of Notch3 aggregates in the brain vasculature, and also the size, as we did for the first study. So we are at the same time also very interested to do the other type of immunotherapy called passive immunization, and this is where we are generating the antibody in the lab, which are more targeting the Notch3 aggregates in the brain. And this is something we also want to see if we can use it as a therapeutic treatment when we have an ongoing pathology, because we believe that this is in most cases, I think the patients, maybe they don't start to have the symptoms maybe when they don't have any Notch3 aggregates. So when they come to the clinic, we believe that this is when we can start, the earliest time point we can start to have this. But we want to make sure that this antibody is, of course, very efficient as well, and also don't give any side effects. So we will use the same mouse model as we did for the active immunization, the more severe mouse model, and we want to start when they are around 2-3 months. But of course, we have to generate the antibody first. So what we have done is that we have used the same protein components, this aggregated Notch3, which we have purified, and we have immunized 5 mice. And the hybridoma technology which we have used is that we take out the spleens from the mice and we fuse them with other cells. So these B cells which are in the spleens, they are the ones who are producing antibodies. So when we fuse them to a myeloma cell line, they get immortalized, so we can just culture them forever. And they can also divide, and we can then see if we can select and single out specific hybridoma cells which are only producing antibody, which are only targeting the aggregated Notch3 and not the normal Notch3. So we have immunized, as I said, 5 mice models, CADASIL mice, and they are here shown in blue. They're called mouse 13.1 to 13.5, and we just looked how much antibody they produce, these hybridomas, and we have looked for if they produce antibodies against aggregates, but also to monomers, which have a mutation, but also monomers which do not have a mutation. And as you can see, they are very well producers, all of them. So we selected just 2 of the mice, and we took out spleens, and we have generated hybridoma clones. We have selected 41 clones, and we have also now done binding tests to see which of these clones prefer to bind to the aggregates, which are these in the green bars, and the ones which are binding to the monomers. So we favor the ones which are binding to the aggregates. And as you can see, most of the hybridoma clones here, they actually favor to bind to the aggregates rather to the monomers, which was very, very positive. So from this 20, we have now selected 10 of these antibodies, and we have cultured them in and purified them to more a scale where we can use it to look if these antibodies are staining the NOTCH3 aggregates, first of all in our mouse model, but also soon we will look into different patient tissue to see if they also can bind NOTCH3 aggregates which have different CADASIL mutations. So this is something which are ongoing, and yeah, as we speak, I would say the lab is really busy in, first of all, evaluating the new active immunization 2.0, and also for this new passive immunization, when we can see if we have a antibody which are favoring to bind to the aggregates, we want to start to do some target engagement and more efficacy in the mouse model to see if actually this monoclonal antibody actually passed the blood-brain barrier, because that is a very important thing, and also that it binds to the aggregates and nothing else. So there is a lot of safety things to do first, both in animal models, and if everything looks positive, we of course want to scale up all these things, both the protein for active immunization, but also the antibody to start clinical trials in patients. And this is the last slide. I just want to acknowledge all the work and all the people and all the funding, and especially for cureCADASIL, the ORF Foundation, for the grant, for make it possible to start and to do this work, and also my group, of course, a very nice group and very eager to find the cure for CADASIL, and also for to our collaborators, of course. And thank you for your attention. I'm very happy to take questions.
Well, thank you very much, Helena. This has been great. Let me change. Yeah, that's fine. We leave the slide on for the moment. So questions. We'll ask people to type questions into the Q&A box that should be, and you should have access to, and so there are a couple of questions which jump right into the realm of clinical trials, I believe, but they're very interesting. I think you no doubt have thought of these things. One of the questions, if given immunotherapy, how often does immunization need to be repeated? And will there be any like age limits? So this is getting over into the clinical realm, I believe, but maybe you could speculate on an answer for that.
Yes, a very important question, because I mean there are differences between this active immunization and also the passive immunization. Active immunization, we believe that of course we need to maybe do it a couple of times in order to evoke immune response in the body. For our work in the mouse model, we did it maybe 8 to 9 times. However, we saw that the immune response was quite stable after maybe half of the time. So I think that we didn't really need to do it as many times. We just wanted to make sure. And in the clinic or in the patients, I think we can maybe think that this is similar to what we need to do for, I mean, COVID vaccine. We may need to repeat it a couple of times to just have an evoked immune response. So for active immunization, maybe not the whole life. For passive immunization, that is, I think, a different story, and the lessons learned from the antibody against the amyloid aggregates in Alzheimer's disease patients. We know that they are doing this every second or every third week. They come into the hospital to get these infusions. So that is how the clinical trial was set up. However, of course, there are now, I think, second generation antibodies in the pipeline to see if the antibody can be more not degraded as rapidly, and it can stay more in the circulation and in the brain. So hopefully, maybe not as often as now, but more than the active immunization.
There are several questions here that are anticipated, I believe, that from patients who want to know, well, when is this going to move into the clinical realm? And I'll give you a couple examples, like how do I register for a clinical trial, and how long does it generally take to get to the stage where patients can go into clinical trials? And so obviously there's eagerness, and I have a question here that Jane Gunther actually had put together that I think kind of summarizes some of this, and that her question was, are there key hurdles to moving your antibody therapy from preclinical models to human trials? And are there biomarkers or other earlier indicators that may help assess effectiveness? And then finally, how can cureCADASIL community help?
Yeah, so of course, yes, we need to do quite a lot of work in preclinical trials and in the mouse models. First of all, in the mouse models, but also in higher species. But that's more to do about the safety of the therapy. We really want to know that this is actually doing what it's supposed to do and not give any severe side effects. And then moving into the clinic, then we of course need to, except for for resources, I think we also need to make sure that we have a good patient group, and which we can give the antibody, or in the other case, active immunization to. And in order to know that, we are collaborating a lot with, we have a European consortium, and we also have one here, of course, at Karolinska, patients who come to the clinic and which we follow, both on with blood clinical data from the blood, but also MRI and different things, so we can also understand which patients would be benefit with this immunization therapies, and which we may have to exclude. So I think for what the CADASIL community could really help us with is actually to be in this type of studies where you can provide your blood samples or maybe do an MRI, so we can understand for each individual, actually, because I think that's also something we have to take in consideration, but because every individual is unique, so we want really to know what stage and when is the good time to give the immunotherapy to the patient.
Well, that's a good segue to another question. That do you envision this? Where in the natural history of CADASIL do you envision this possibly being administered? Would it be preventive? If one could identify that, the problem, of course, being the heterogeneity of the disease is so great. But would it be possibly preventive, or would it be early stages? I know these are impossible questions, probably, but
yeah, I think we may maybe look into what has been done for other diseases, and then I give the same example for Alzheimer's disease, which is which has now been it's ongoing. And I think what we can see there is that the earlier we can come in, the better. However, I think to go for preventive before, I think that can be also some ethical issues about that, because we don't really know when it's going to happen, although we may know that it's going to happen, but not really when. But also for safety reasons, I think early stages, I think, would be preferable.
There's another question that had to do with the variant locus, if that would influence therapy. Because as you know, the EGFR is different in terms of risk prediction and whatnot, and if there are any inclinations there.
So what we have done in our both the active and the passive immunization, that we have used a short version of the Notch3 protein, which have the majority of the CADASIL mutation, not all of them, but the most of the mutations are within this EGF 1, 2, 5, 6 repeats. So we have one mutation in our protein which we have used for immunotherapy and also to have as a target for generating the antibodies. What we can see is that we have one mutation in the protein, but we are doing it in a mouse model with a different mutation. So, and it seems to work. So we believe that we can hopefully have a more generalized immunotherapy, both for the active and for the passive immunization with the mutation carriers, with different mutation carriers. But of course, we want to test that in the lab first to see if there are any selectivity towards certain mutations.
I have to read you this comment because it does reflect, I think, some of the other patients' comments as well. That thank you so much for your dedication to research a treatment that hopefully one day will help our loved ones. Where can we follow your findings?
So as an academic researcher, I am publishing my results, and it should be open for everyone to read. And of course, I think if we find a cure or something which we want to use in the clinic, I think that will be something which will be spread through the CADASIL community quite rapidly. And of course, then it's up to each country, of course, to decide if they want to be part of different trials and so on. But I think it will be spread.
Yeah. But there's another question here too: is what is the outlook for continued funding of this research?
Yes, that's a good question. So the job for a researcher is to apply for funding all the time. So I have funding, of course, from cureCADASIL and also from different brain foundations and also for European Union. We have a big consortium of 6 different sites in Europe where we collect clinical data, we collect blood, MRI images, but also we do a lot of studies in both mouse models, but in also patient tissue from patients to understand if there are differences between the different mutation carriers, of course, depending on where the location of mutation is, to maybe know more about subgroups which we can use for the future therapy treatment. So yes, we are working with funding, applying for funding all the time. But I think the more positive results you get, of course, funders are more willing to give support.
I have one question that I think some others have shared a bit on too, and that's coming back to biomarkers. Recently results have been published about skin biopsy, for example, being correlating very highly with clinical findings. What do you see a biomarker? I think that obviously it would be great if you could do plasma levels or some such, but that may be a little little hard to do, in a clinical setting to be predictive enough to use as a biomarker. And so I'm wondering if you've obviously given thoughts to that. I know there's probably not yet enough data on that, but what is your take on what might end up being a clinical biomarker if one were to have a clinical trial set up?
Mm. So I think we are right now going quite broad to look into, as you said, the skin biopsies, but also blood biomarkers. We have also the spinal fluid, which we can also detect proteins in. And that's maybe a bit more invasive and not so common in every country to do. But also for MRI, to look for changes quite early on. And I think right now, I think some imaging biomarkers are quite promising, but of course you need to go to a specialist clinic to do that, and that can be maybe cumbersome, and of course resources taken from that. But I think I give my hopes quite a lot to finding blood biomarkers, because in the end we have a disease which are affecting the vasculature in the brain. So I think we can find key proteins there which are maybe changed quite early in the disease onset as well, where we can find good biomarkers where we can see disease progression, and also to measure the therapeutic efficacy and so on.
Great. I'm going to switch screens here. And I want to thank you very much. This was a great presentation. It's very encouraging to hear. I think it's an important path to continue down to look for potential therapies. And I wanted to thank our audience too. They've had great questions, and I know a number couldn't make it who will be listening to the recording. And so we have a recording of this webinar, and it will be available. It usually only takes a couple of days, and it will be available online. You can go to our cureCADASIL.org and online and look for the webinar. And also look out for our future webinars that we will bring to you. And also we're having a patient investigator meeting coming up on June 29th, where we'll be looking at a number of investigations that are going on with CADASIL. And so you can sign up for that if you wish at the United Leukodystrophy Foundation website. So anyway, those are our advertisements. And again, thank you, Helena and everyone. And we hope to look for your results in the future. Thank you.
Thank you. And I'm happy to come back and give more update on the ongoing studies.
Great. Thank you.
Thank you, Helena. That was wonderful.
Yeah, I think she missed out. That was fabulous. That was fabulous. I am so thrilled, and gee, if there's anything we can do to accelerate her work, because this does sound promising.
Yeah, you know, we could remind her that she can reapply for the $1 Million Bike Ride grant. She may not know that we've given the grant out twice to Fabrice. So, you know, if her research is the best one to put our research dollars towards, I'd love to see her apply again.
Yeah. Okay, Jane. Thanks.
Yeah, thank you all for all your work, Anita.
Pleasure.
This month is a big one.
Yeah, I think we may be on microphone for everybody. So we'll talk to you later.
All right, bye. Hmm.